What I did was I went from the way we've been talking about dynamic programming with the alignment graph to formalizing that in terms of notation. So the basic ideas are already there. All you have to do really is to see that we've taken what was a graphical view and turned it into something notational. And the notation is for two reasons. One is because you can't hand a picture to a computer and say, run with this, but you have to give a program. Programs are in terms of text and notation. So the notation got us a lot closer to the program. And in fact, the Needleman program on your lab this week is just a translation of those recurrences that I talked about. And the second reason to do this is because to introduce you more generally to dynamic programming, which is not always so nicely represented in these graphical terms that alignment was. Okay, so let me, for those of you who were not here, the notes of what I talked about last time are on the web, exactly what I displayed, and you should take a look at that. What I will do today, sort of to emphasize what I talked about, was to talk about the recurrences in more generality, in terms of substitution matrix. And so let me find where that is. Okay, so I'm going to, actually let me put this up on the board. It's a little bit, I think, easier for people to absorb when I take the time to write it out as opposed to just putting it down there on the overhead. So let's imagine from a substitution matrix when you've aligned, you have an alignment, and you have a character in position I of S1 and a character in position 2, or a generalized character, so it may also be a space, in position J of S2, the value, which may be negative, in which case you think of it as a penalty, but the general term is value or score. I'm going to denote that as S. If I had my glasses on, I could actually read this. But S1 of I and S2 of J. Okay? So we're at position I, but that's not of the original sequence S1. That's I after you put in the spaces. So at position I in the alignment, and this is position J in the alignment, and here we have position J of S2. Okay, so remember okay. A bit of a let me change these notations just for some convenience. L and K. I just want to use different subscripts than what I'm going to do in a minute. Okay, so this is where we take the Lth character in the alignment and the kth character in S2, and we look up in the scoring matrix, little s, what their values are. Now, this is what's important. Vij is defined as the maximum alignment score obtainable if when we only use characters 1 through I of S1 and characters 1 through J of S2. Another way of saying it, and the way we said it last time, was that this is defined as the similarity value of the prefix of S1 consisting of the first I characters and the prefix of S2 consisting of the first J characters. So another way of saying Vij is the similarity value of the prefix S1, 1 through I, and S2, 1 through J. This is just notation, okay? And then what we want are recurrences that explain how you get this by making reference to values of V for smaller indices I and J. Okay? So the base cases, V0J. This is saying we're going to use nothing of the first sequence, but we're going to use the first J characters of the second sequence. So what does the alignment look like in terms of those first J characters? What is opposite those first J characters of S2? If you're using nothing of S1 and J characters of S2, what's the alignment look like? It's what?
Because it's a space opposite character. So
A space opposite, yeah. So what you'll have is a space opposite each character. So we'll have up to the jth character, whatever this happens to be, A, W, X, Y. Here's the jth character in S2. It's being used. There's nothing being used from S1. So everything, the first J characters are opposite of space. And that means that this value is equal to K less than or equal to J, greater than or equal to 1, S with a space, and S2K. Okay? So what you're saying, what this is just simply saying is, whatever that character is that's in the kth position of this alignment now, opposite of space, what do you get from the substitution matrix? The substitution matrix may be such that every character opposite of space gets the same penalty, but this is a more general notation that allows a different number even for each particular character. So this says, depending on what that character is opposite of space, we're going to get some particular value, and we're just going to add them up. And that's the score or the value you get from for V0J. Similarly, VI0, by the same reasoning, K less than or equal to I, greater than or equal to 0, S1K space. Okay? So if you have the first K characters of S1 placed opposite spaces, then the value you get for that is this summation. And then the more general recurrences, Vij. Well, last time we related this to the different cases of what happens to characters I and characters J, character I and J. And well, we have, it's the maximum, it's the best that's possible over basically 4 different possibilities. Okay? Vi minus 1, J minus 1. That means we're going to align the first I minus 1 characters of S1 with the first J minus 1 characters of S2 in the best possible way. That's what you get for V. And that means that the ith character, that the two characters Ij are going to be next lined up with each other. Okay? So what does that What value do you get for that? What? Well, okay, previously it was +1 if they agreed and it was -1 if they disagreed. Now we're doing something more general. We're going to take that value from the substitution matrix. Okay? So it's going to be whatever the ith character is of S1 and whatever the jth character is of S2. Okay, these two particular characters, if they're lined up, if they're aligned, then we take that particular value from the substitution matrix. And what you can see is that this notation covers both matching and mismatching cases. Okay? Or we have V I minus 1 J plus S1I with a space. That is the ith character of S1 opposite a space. Or we have I J minus 1 plus S2 J opposite a space. Okay? So these are the three possibilities. And I just wanted to point out, this is the generalization of the recurrences we did last time, where this was either a plus or -1, depending on whether these two characters matched. This was always a -1, and this was always a -1. But otherwise, really the logic of these are exactly the same. And how we go about computing the similarity value is the same as before. Okay. Now I will just throw up one of these tables. This particular table comes from back to the case where we're adding +1 for a match and -1 for a mismatch and so on. But I just want to emphasize the order in which you do this. This happens to be alignment of vintner and writers, two particularly common words. And what I did was already put in the 0 column and 0 row. But again, this is the with the -1 for each mismatch and a -1 for each space. And just like we did when we had an alignment graph, you would, after you fill in those, you would fill in that cell, then you could fill in that cell, then you could fill in that cell, and so on. And when you finally get down to this cell, you have the similarity value of those two sequences. So here's a next intermediate step of that. You see, if many values were already in here, we're starting to work on this row, and this is the next one that's available to be filled in. And in the case of the original objective function where you have +1 for match, -1 for mismatch, and so on, you would just look at the three neighbors and the two characters T versus T. That happens to be a match. And now you would consider -1 with a +1, which is a -1. Sorry, -2 with a +1, which is a -1. Or -3 with a -1, which is -4. Or -2 with a -1, which is -3. And the best would be -1. However, now with the general recurrences, you would have to look up what do you get from a scoring matrix for a T against a T. And that would be -1 plus whatever that is you get from the scoring matrix. And then you would have to say, what do you get for a T opposite a space? And then whatever that happens to be would be -3 plus that. Here is also a T opposite a space would be -1 plus that. So I know I'm going through very fast. That's because I did this basically last time, and because it's got the same logic of what we were doing when we had a alignment graph. But I'm now pointing out that you do these computations typically on a table, and in a programming language that means an array, and you do them with these more general recurrences, which means notation and it means a substitution matrix as opposed to just a uniform score for each match and mismatch. But otherwise, the logic is really the same. And more important, because the logic is the same, because when you fill in a cell, you're only looking at its 3 neighbors, plus maybe looking up something from a table. You still only have a constant number of operations per cell, which means that the amount of work, the number of operations that you do to fill in this kind of table with the general recurrences is still proportional to the product of this length times that length. It's the product of the lengths of the 2 sequences, even though we have a more complex objective function now. Okay? And that's worth pointing out, so that we can do generalized alignment with these general recurrences almost as efficiently. The only thing that changes is the constant of proportionality. We have to look up a number from a substitution matrix, several of them maybe, for each operation. But the total number of operations is still proportional. The constant of proportionality may change, but it's still proportional to the product of these 2 lengths. And that means it's still very, very efficient. 2 sequences of length 1,000, product is 1,000,000. Take 20 operations for each cell if you like. That's 20 million. It's no time at all in a modern computer. Okay. So that's what I'm going to say about that, which really now makes us ready for new territory. I guess the only thing I want to say about this is that this is called global alignment because we use all the characters of both sequences. So we're aligning the entire intervals of both sequences. And this is also sometimes referred to as Needleman-Wunsch alignment. Well, this is somewhat of a misnomer because Needleman-Wunsch focused on this problem and gave an algorithm for it. However, the algorithm is not exactly what we're using today. The algorithm they proposed was not quite as efficient as the one that we presently use. But nonetheless, the name has stuck. Needleman-Wunsch is oftentimes referred to when you're talking about global alignment. All right. And now what I want to do is shift gears to a different kind of alignment called local alignment. So global alignment, I just told you, has to do with aligning all characters in both sequences. Local alignment is motivated by the following picture. Let me see if I can find this quickly. Yep. So local alignment is motivated by the following picture in the following situation. You may have 2 very long sequences, DNA or protein, which have some regions of high similarity. And by high similarity, I mean by exactly the definition of similarity that we've been discussing. So that if you were to take this substring and align it with this substring by global alignment, but global only using this one and this one, you would have a very high similarity value, high relative to the 2 lengths, let's say. But if you did a global alignment of all of this with all of this, it would not look particularly similar. These 2 sequences would not be highly similar. And this is a situation that happens very, very frequently in dealing with molecular sequences, both DNA and protein sequences and RNA, and for many different reasons. Okay? Let's just take DNA. Can anybody propose a reason why you might have some subinterval with very high similarity, but the rest of it is quite different. I know you're all working, molecular biology labs, and you probably have many reasons. Somebody throw out one, at least one.
Promoter. They share the same promoter shape, same motor part. So, I don't know.
Okay, the key word in what was suggested here was promoter. Okay, it's certainly very common that two different genes have very similar promoters. Sequences. And that may, in fact, have some functional importance. But the point is that you may have a sequence here that has a gene and a sequence here with a very different gene. So this is gene A and this is gene B. But the promoters are virtually the same. And that may have some historical meaning or some functional meaning. It's not clear, but if you took a whole subinterval of DNA here and here, it may very well be that it's just these promoters that have high similarity. And in fact, it may be that that's why you're trying to align them, is to find the promoters. You're trying to find out what are the regions, the subregions of similarity, so that you can say, yeah, I think this is a promoter. Well, another possibility is that you may think that you have very similar genes there. So gene A and gene A prime, but these are very similar genes. When does that happen? You grab a hunk of DNA from a fruit fly, 20 million bases, and you think there's a gene in there for some particular function, and you grab another hunk from a cow or a human or whatever, and you think there's a similar gene as the same function, and as we discussed in the very first lecture, the leverage we're trying to use here in trying to identify genes that have similar function is by finding ones that have similar sequence. But all the other stuff may be quite different. The order in which the genes that surround it or the DNA between genes may be quite different. But you don't know where that gene is. You don't know where GA and GA prime is, but it's somewhere in 20 million base pairs. So you align those 20 million, and you try to find highly similar regions rather than trying to align all the characters in one with the characters in the other, because mostly that would just give you garbage, okay? Because mostly they're not similar at all. So that's another case where local alignment comes up in DNA. You could probably write down, it wouldn't be difficult to write down 50 different ways in which local alignment arises in DNA. What about protein? Well, to some extent, phenomena in protein just reflect what's happening in DNA. But just thinking more in terms of protein and protein evolution, what can you say that would suggest how local alignment comes about? What? Sorry, say it again.
Active sites, binding sites of protein should be kept similar in different species.
Okay, yeah. So that was a suggestion that and we talked about that actually the first day, that the functional regions of a protein, and what was mentioned there was binding sites, are more highly conserved than the non-functional parts of the protein sequence. So you may have back at some point in time a divergence, whether this is an orthologous or paralogous divergence, doesn't really matter. You may have two protein sequences now that had some common origin, and they're mutating, but what's remaining more constant and conserved are the functional parts, such as the binding, I've forgotten what you called it, but anyway, binding region. So if you were to align these two sequences, you may find only a small part of them are highly similar. And in fact, that's what you want to find, because that would then tell you what regions you think are the most important functionally. And people do that all the time when they don't know they don't really understand the protein very well, but they want to have some hint as to what are the most important functional sites. Okay? The rest of it can be have drifted apart. There's a certain art to this, by the way, because if you pick two species here that are not very highly diverged, then you're going to see a lot of similarity just because it hasn't been very long. If you wait too long and the sequences are tremendously diverged, then you may not be able to see what's being conserved. Even that may have mutated as well, because there is some rate of mutation. So you want to pick your two sequences at sort of the intermediate stages where you're likely to see what's conserved, where the non-functional part will have drifted away, but the functional part will more or less be conserved. So that's one story in protein. What's another story in protein? Well, let me give you a quote here about sort of the dominant view of protein evolution. Actually, I think I mentioned this. I didn't give you this quote, but I mentioned this the first lecture or so. This is a quote by Cyrus Chothia, who's a well-known protein structure chemist, evolutionary researcher in protein evolution as well. Extant proteins have been produced from the original set, whatever they are, not just by point mutations. We've been talking about point mutations mostly. That's the single character mutation, a mismatch. Not just by point mutations, insertions, and deletions, but also by combinations of genes to give chimeric proteins. This is particularly true of very large proteins produced in the recent stages of evolution. Many of these are built from different combinations of protein domains that have been selected from a relatively small repertoire. So the key word here is protein domains. You think of long proteins, or what's stated in that quote, proteins that have been produced recently, most recently, as being produced from a set of Legos. And you've got these basic little Lego pieces in large quantity in a container, and you can reach in and pick out a few and stick them together, and that's a protein. Or you can pull out another one and stick them together. That's a different protein. They may share one or two of those Lego pieces. And the Lego pieces, protein domains, are relatively large. But the point is the rest of it, because it's different Lego pieces, is going to be different. So you may have this chimeric protein consisting of these protein domains. And here is a blue one, a blue Lego piece, and here's another blue Lego piece. And the rest are all different. Okay? And so the only important similarity, or only significant similarity you're going to find is this one protein domain. The reasons for why you want to find that and see that are quite varied. Anyway, many, many, many different reasons for local alignment. And when it comes to protein, your protein sequences, you more typically want to do local alignment to find these regions of local similarity than you want to do global alignment. Global alignment comes up frequently in evolutionary studies where you think that two sequences you already have some reason to believe that they're reasonably similar throughout their entire lengths, and then you would do global alignment. But local alignment is probably more common in bioinformatics than global alignment is. So how do we formalize? We formalized global alignment before. Let me try to formalize local alignment. Okay. So given S1 of length n and S2 of length m, what you want to do is find a substring alpha of S1. And a substring, by the way, is contiguous. You have a long S1. A substring is from position X to position Y. You get to pick X and Y. It's not that this character, and then a little bit later some other character, and a little bit later some other character. That's called a subsequence. But a substring is contiguous. So you want to find a substring alpha of S1 and a substring beta of S2. Okay? So that V alpha beta, that's the global alignment value. The global alignment value of alpha and beta. Okay, so that V alpha beta is the largest V value over all pairs of substrings. Of S1 and S2. So conceptually, if I have all of S1, I have all of S2, this definition says, imagine taking every possible pair of substrings from S1, one from S1 and one from S2. And for each one of those, compute the V value of those two substrings. And then for all of those, show me the one that has the largest V value. And that is what's considered the local alignment. That is considered the two regions of interest, one from S1 and one from S2. And of course, the global alignment of those two would then be displayed. Everybody with me? Or this is, I know, abstract. Yeah, question. Good.
The length alpha and beta are determined by yourself or during the calculation.
Okay, the question is, what's the length of alpha and beta? Alpha and beta here, in the end, are some specific string. This is a specific string alpha, substring of S1, a specific string beta, which is a substring of S2. But a priori, we don't know anything about their lengths, except, of course, they're no bigger. Alpha is no bigger than S1, and beta is no bigger than S2. The definition says, think of all possible strings in S1 and all substrings in S1. Think of all possible substrings in S2 and all possible pairs between them. Then, for each one of those pairs, compute the V value. And the V value, which is the largest, that's our guy. Okay? Did that answer your question about lengths? Yeah.
Is the space totally not allowed at all, or
is what?
The space, it has to be absolutely continuous or not?
The question is there a space allowed in the alignment of alpha and beta?
Right.
What's the answer?
Yes.
Yes. Alpha and beta as substrings. Alpha is contiguous, beta is contiguous. But the alignment of alpha and beta can have spaces. That's because we're talking here about V. This is the value of this is the global alignment value. Global alignment allows spaces. Okay? So we're not just talking about an alpha which is has to be compared to beta without spaces. The comparison of alpha to beta can have spaces in it, definitely. Okay? So let's maybe to make this more concrete, let's try to do some counting. Based on the definition, if you were just to write a program based on the definition, or again, this is my thought experiment. Somebody comes in and says you have 5 minutes to write a program that does this. The program doesn't have to be very fast, but the programming has to be fast. Okay, and if you can write a program that works that is correct, and you've done it in 5 minutes, you get $1 million. If you don't, you get shot. So what's the naive way of doing this, the really straightforward way of doing this, and it's related to counting? You would, of course, just generate every possible substring in V1, in S1. And you would generate every possible substring in S2. And you would generate every pair of those possible substrings. And so those are just loops, right? Looping through some programs. Looping through some program. And for each pair of substrings, you would then call a subroutine that computes V alpha beta. We even have that. It's the Needleman program. And you get back the value, and you look to see whether it's bigger or smaller than the previous best, previous largest, and you would accumulate the largest as you go. Okay? I could ask you in this approach how many loops it takes, but implicitly that's what we're going to do when we count. So how many substrings of S1? How many substrings of S1 are there? S1, remember, has length n. It's what? One substring of S1?
Depends on how long the substring is.
Well, yeah, it depends on how long it is. How many substrings of length n? One. How many substrings of length 2?
2.
2. How many substrings of length 3? 3. How many substrings of length 4? How many people are tired of this? How many substrings of length I? I. Okay? So summation I equals 1 through n. Okay? That's the number of substrings there are. And how many, who knows what this summation is? Everybody learned this in high school, and you used it in your statistics course or elsewhere, I'm sure.
N times n plus 1 divided by 2.
Yeah, it's n times n plus 1 divided by 2. And I'm just going to call this n squared over 2, okay? It's about n squared over 2. And when we're in the frame of mind where we're throwing away constants, as we're going to be talking about proportionalities, we can even throw away the half, okay? But so you can see it grows as n squared. As the sequence gets longer, the number of substrings grows proportional to n squared. Okay? And how many substrings of S2? How many? Well, what do we know about the length of S2? It's length m. Okay? So that's about m squared over 2. All right? So we've got n squared, roughly, substrings of S1. We've got m squared of S2. Yeah, question?
I don't know substring of what?
Yeah. There's n substrings of length 1. N substrings of length 1. And 1 substring of length n, and I substrings of length I. So the point is that the total number is just the summation of the integers from 1 to n. What's confusing about this is this looks like 1 plus 2 plus 3, and if you think of the first number as being the number of substrings there should be of length 1, then that looks backwards. Everything got reversed. But it's the same sum. However you think about it, in the end, it's this sum. By the way, a quicker way of thinking about all this is just to say, how do you describe a substring? You pick a first point, and you pick a second point. So you pick 2 points, or 2 indices in that string of length n. And how many pairs are there? About n squared. Okay? Well, you didn't really want to pick the second one before the first one, so that divides it in half. Okay? That's just sort of a hand-waving way of, you'd get to the same idea here that's about n squared substrings. Okay, so let me go over here. So how many pairs of substrings? One from S1 and one from S2.
Multiply the 2 numbers.
Multiply the 2 numbers, right. N squared times m squared is also, well, it's about, okay, nm squared. Okay? So it looks like a big number. Let's take n be 1,000, m 1,000. Previously we said you can do global alignment of 2 sequences of length 1,000 in almost no time at all. Because 1,000 times 1,000 is only 1,000,000. But 1,000,000 times 1,000,000, now you're talking about real money. That's getting into some big numbers. Okay? Moreover, this is just the number of pairs. For each one of them, you have to do a global alignment. Okay? Now, the cost of each global alignment depends on how long those 2 sequences are. For 2 sequences of length 1, that doesn't cost very much time. 2 sequences of length n, now you're, or 2 entire S1 and S2, now you're talking about a lot. But if I were to write the sum and sum it, does anybody have any idea how much that would be? Well, what's the, let's put it, I'm sorry, what's the average time for alignment over all of those possible pairs? Well, I'll let you do that summation at home, but you don't save much over n times m. Okay? When I take all of S1 and all of S2 and I align those, that's going to be n times m operations. That's global alignment of those 2 sequences. Now, the average over all those pairs, some of whom are small and some are large, I think, is not, it's going to be smaller on average than nm. That's the biggest possibility. But on average, you're still going to be proportional to n times m. So, total number of operations for local alignment. And by that, I mean via the definition. If you just do it the way the definition suggests doing it, is what? It's nm. It's about nm cubed. So when n was 1,000,000, when n was 1,000, nm is, well, n and m are both about 1,000, and this is about 1,000,000. 1,000,000 cubed is a big number. Okay? And people actually want it, will align sequences that are much bigger than millions. Some people want to do billions. So two genomes aligned, billion squared, and then to the third is a billion to the sixth, which is a large number. Okay, so let's say for n roughly m, this is starting to look like n to the sixth. So for global alignment, it just cost us n times m. That was the time it took to compute the global alignment. And the reason I'm emphasizing this is because that made global alignment practical. For local alignment, if you follow just if you do the computation according to the definition, it's starting to look bad. If this is not exponential, you have the n down here and the exponent is a constant. That's better. I mean, this is n6 is a lot smaller than 6 to the n, but this is still very rapidly growing. Okay. So, but I hope in laying out sort of the naive approach here of this counting, it's clarified what the definition of local alignment is. So, does anybody want to ask a question about the definition of local alignment or about this counting? Okay. Now, how many people on XParal, which you all used last week, on the alignment choices, one was global, which is what mostly you used, but another one was local. Did anybody try local alignment on their sequences, on the long sequence? We had sequences of length about 120 or so. 120 to the sixth, I think, would be out of the question. Okay. But it didn't seem, if you played with XParal, and go back and try it, it doesn't seem to take any longer to do local alignment than to do global alignment. And by the way, in XParal, when you do local alignment, you have your two sequences. And in the alignment window, it will show you by square brackets what the interval is, what the alpha and beta essentially is, that it's found. Those are the two sequences where the global alignment between the two is the best overall possible pairs. And so here is the fact. Local alignment can be computed using, again, something proportional to n times m operations only. So in fact, despite what it looks like if you just follow the definition, if you're sufficiently clever, find the right way to do it, you can compute the local alignment according to the definition. You're actually getting the result that the definition specifies, but you're not doing the computation following the way the definition lays it out. But via a more interesting dynamic program than for global alignment, you can find the optimal local alignment in time that's roughly the same as for global alignment. The computational burden is roughly the same. And this result, the method that does this, is called Smith-Waterman. And how many people have heard of Smith-Waterman method in bioinformatics? Okay. So I don't have time today to tell you what the method is. I will eventually tell you the method, and you will have the exercise of taking the global alignment program, the Needleman program, and converting it to a Smith-Waterman program.